Let's start with a pretty brutal, unavoidable reality.
Yeah.
If a patient is diagnosed with pancreatic ductal adenocarcinoma, or PDA, right, they are facing, honestly, one of the deadliest cancers on the planet.
Yeah.
I mean, the 5-year survival rate sits at less than 10%.
It's incredibly grim.
And the biggest hurdle for you, the patient, or for the doctors, isn't just detecting it early. The real nightmare is that these tumors are notoriously just stubbornly resistant to chemotherapy. You know, you can hit them with the most potent drugs we have, and the tumors simply shrug it off.
They just adapt to it.
Right. But what if a tumor's resistance to drugs isn't just, a property of the drug itself? What if it all comes down to this hidden metabolic identity within the cancer cells themselves?
And that right there, that is the paradigm shift we're looking at today. Because, I mean, for decades, oncology has treated tumors based on their surface-level genetic mutations. We read the DNA, we categorize the cancer, and then we treat it based on that.
Right, the standard protocol.
Exactly. But beneath that surface genetics, tumors with the exact same mutations can actually be running completely different internal operating systems.
Which is wild to think about. So, welcome to this deep dive, everyone. Today, we are exploring a really major scientific breakthrough. This is detailed in a July 2026 paper published in Cell Death and Disease by Wang and colleagues.
A fascinating read.
Oh, absolutely. And our mission today is to unpack the biology of a specific protein called LSD1. We're going to see how this single genetic switch can either make a tumor incredibly vulnerable to chemotherapy or, and this is the crazy part, make it practically invincible.
Yeah, and it all depends on the tumor's internal engine.
Exactly. So let's establish our main suspect here. What exactly is LSD1? I mean, break it down for us.
Right. So LSD1 stands for lysine-specific demethylase 1. It is what we call an epigenetic regulator.
Epigenetic. Let's make sure we define that clearly.
Sure. So to understand that, think of DNA as the master blueprint of a house. Like, the blueprint itself doesn't change, right? Epigenetics is like the contractor reading the blueprint. They're the ones deciding which rooms to build, which rooms to ignore entirely.
Oh, I like that analogy. So LSD1 is the contractor.
Exactly. It operates by removing these tiny chemical tags called methyl groups from histones. Now, histones are the proteins that your DNA wraps around, sort of like thread around a spool.
Okay, got it. Spools of thread.
Right. And by removing those chemical tags, LSD1 basically dictates how tightly the DNA is wound up on that spool, which, in turn controls which genes are actually accessible to be turned on or off.
And in the context of this deadly pancreatic cancer we're talking about, what is LSD1 doing on the job site?
It is heavily, heavily overexpressed.
Like too much of it.
Way too much. The tumor cells are producing massive amounts of it. Clinically speaking, when a pathologist looks at a pediatric biopsy and sees high levels of LSD1, it points to a very, very poor prognosis. It drives faster tumor growth, more aggressive spreading, and generally just a much nastier phenotype all around.
Okay, I have to stop you there, though, because the logic, it seems almost too simple, right?
How so?
If LSD1 is this master switch driving aggressive cancer, then shutting it off should just universally cripple the tumor. I mean, it's like finding the main breaker box to a villain's secret lair. You pull the lever down, the power goes out, the lair goes dark, and then the chemotherapy sweeps in and finishes the job.
And you know what? The researchers shared that exact hypothesis. That's exactly what they thought. They assumed silencing LSD1 would be a universal vulnerability across the board.
So what do they do to test that?
Well, they took different pancreatic cancer cell lines in the lab. And for our purposes today, we can kind of group these into two main metabolic categories.
Okay, what are the two groups?
First, you have the RSK-subtype. This is characterized by certain growth signaling pathways. And second, you have the KRAS-subtype, which is driven by, one of the most notorious mutated oncogenes in all of cancer biology.
The KRAS mutation, right?
Yeah. So the researchers used genetic tools to basically knock down or drastically reduce the levels of LSD1 in all of these cells, turn off the breaker box, like you said. Then they hit them with standard chemotherapy drugs like gemcitabine and paclitaxel.
Okay, and let me guess. The breaker box analogy didn't hold up in the real world.
It only held up half the time.
Yeah.
And this right here is where the paper throws a massive curveball.
Okay, lay it on me.
In the RSK-subtype cells, knocking down LSD1 did exactly what you just described. It sensitized the cells. The chemotherapy just tore through them, inducing massive cell death.
Okay, so that worked.
And here's the twist in the KRAS-subtype cells, knocking down that exact same protein induced profound resistance. The chemotherapy suddenly became highly ineffective.
Wait, hold on. That makes absolutely no sense on the surface. We're talking about the exact same genetic intervention here.
The exact same one.
You silence the same bad guy protein, and half the cancers get weaker, while the other half somehow become bulletproof against the exact same chemo drugs.
It's wild, right? The divergence is just staggering. And I think it highlights why pancreatic cancer is so incredibly difficult to treat in the clinic.
Because it's a moving target.
Exactly. You have the same disease, the same targeted therapy, but entirely opposite responses from the tumor. So the researchers realized that to answer why this happens, just reading the DNA wasn't going to be enough.
Right, they had to go deeper.
They had to look into the physical architecture of the cells themselves. Specifically, they had to look at how these cells generate their power.
Okay, let's do a quick recap for the listener, just to keep everything straight. We have a protein, LSD1, turn it off, and tumor type A, the RSK cells, becomes weak. But tumor type B, the KRAS cells, becomes super resistant.
Perfect summary.
And the mystery lies in the power grid. So we really need to talk about mitochondria.
Yes, the mitochondria. As we all learned in high school biology, they are the powerhouses, or the engines of the cell.
Right.
When Wang's team actually analyzed the mitochondria in these two different subtypes, they found two completely distinct worlds. So let's look at the RSK subtype first, the ones that became vulnerable to the chemo.
Okay. What do their engines look like?
Their mitochondria are beautifully functional. They perform a process called oxidative phosphorylation. We usually just abbreviate it as OSPHOS with incredible efficiency.
OXPHOS, meaning they use oxygen to burn fuel and create energy, right? Which is how a healthy, normal cell is supposed to operate.
Correct. It's a clean, high-functioning factory floor. But, maintaining a clean factory requires a really robust waste management system.
Sure, things break down.
Exactly. So these RSK cells have this highly active recycling system called mitophagy. The prefix mito for mitochondria and phagy meaning to eat.
Right, to consume.
So when a mitochondrion gets old or damaged or just inefficient, a specific protein called BNIP3 steps in.
BNIP3.
Yeah, BNIP3. It basically flags the bad engine. It puts a tag on it. The cell then engulfs that damaged engine and breaks it down for spare parts, keeping the factory floor pristine.
So the RSK-subtype is this pristine factory. The machinery works perfectly, and if a machine does break, this BNIP3 protein flags it, and maintenance clears it off the floor immediately.
Exactly.
So what does the KRAS-subtype factory look like, the one that gets resistant?
It is a total disaster area. I mean, the KRAS cells suffer from profound mitochondrial dysfunction. We're talking about severe structural problems here.
Like what kind of problems?
Well, they have mutated mitochondrial DNA, specifically affecting something called complex I. Now, complex I is the very first gate in the electron transport chain. It's basically the primary turbine that starts the whole energy generation process.
Oh, wow. So the very first step is broken.
Right. And because this primary turbine is broken, their biochemical balances are completely thrown off across the board.
When you say biochemical balances, what are we actually measuring there?
The researchers measured the ratio of NADH to NAD plus S.
Okay, let's break that down for everyone listening.
Sure. You can think of NAD plus as an empty battery, and NADH is a charged battery that's carrying electrons. Now, in a healthy cell, electrons are constantly being passed down the chain to make energy, so that ratio stays balanced.
But in the broken factory?
In these KRAS cells, because complex I is broken, those electrons just get backed up. They can't move forward. So the battery ratio just drops drastically. But, and this is the most critical failure of all, they lack that BNIP3 protein we talked about.
Wait, so the waste management system is offline?
Entirely offline. They cannot recycle their bad mitochondria. If you look at these KRAS cells under an electron microscope, you do not see clean engines.
What do you see?
You see a crowded, just disorganized mess of defective, swollen mitochondria literally piling up inside the cell. They can't function, and they can't be cleared away.
Oh, man. So imagine a massive factory assembly line. The RSK factory is pristine. Raw materials come in, the conveyor belts run smoothly, and finished products go out. If a belt snaps, the maintenance crew instantly removes it.
Beautiful.
But the KRAS factory, on the other hand, is a nightmare. The main conveyor belt is jammed at step 1, sparks are flying everywhere, the floor is covered in broken, swollen machinery, and the maintenance crew has basically quit.
That is exactly what it looks like. And You know, we typically classify a patient's cancer by its genetic mutations, like saying, oh, you have a KRAS mutation.
Right, that's what the chart says.
But this paper is suggesting that a tumor's physical metabolic health, the actual state of this factory floor, is just as critical in determining how it reacts to stress like chemotherapy.
The state of the factory floor dictates everything.
Exactly. And this brings us back to our epigenetic switch, LSD1. Because you might be wondering, how does shutting off LSD1 actually connect to these two very different factories?
I was just going to ask that. How do they link up?
The researchers found the missing link. It's an enzyme called GLS2.
Okay, GLS2. Let's define that one clearly.
It stands for liver-type glutamate. Now, normally, LSD1 acts as a repressor for the GLS2 gene. It basically sits on the DNA and keeps the production of this enzyme very, very low.
So it's keeping the brakes on.
Yes. But when the researchers knocked out LSD1, they lifted that repression, they took the brakes off. The result was this massive spike in GLS2 levels inside the cells.
And what does GLS2 actually do on this factory floor?
Its main job is to process an amino acid called glutamine. Glutamine is highly abundant in the human body, right?
Yeah.
Well, GLS2 takes that glutamine, converts it into a specific type of fuel, and feeds it directly into the cell's mitochondrial engine into a pathway called the TCA cycle.
Okay, I follow you.
So by knocking out LSD1, the researchers essentially opened a floodgate. They started dumping massive amounts of glutamine raw material directly into the mitochondria.
Okay, I need to challenge this because mechanically, this is where the puzzle gets super confusing for me.
Go for it.
You were telling me that turning off LSD1 floods both factories with the exact same raw material, this glutamine.
Exactly the same, yes.
In the pristine RSK factory, this flood of material makes the cell die when chemotherapy hits. But in the KRAS factory, which we just established, is completely broken, filled with trash, and has jammed conveyor belts. This flood of material somehow makes it stronger.
It does.
But if you flood a broken, jammed factory with a massive surplus of raw materials, it shouldn't get stronger. I mean, logically, it should just jam completely and collapse under the weight of its own toxicity.
Yeah.
How does a dying cell use a flood of raw materials to survive chemo?
That is a brilliant question. And to answer that, the researchers had to actually track the fuel. They use a technique called stable isotope tracing.
Oh, this sounds cool. How does that work?
It's fascinating. They took the glutamine fuel and replaced its normal carbon atoms with carbon-13, which is a heavy isotope.
Heavy meaning literally heavier in weight.
Yes, physically heavier than normal carbon. So scientists can use a mass spectrometer to literally weigh the molecules inside the cell. It allows them to trace exactly where the carbon atoms from that specific glutamine end up step by step as they move through the metabolic pathways.
So they essentially painted the raw materials neon green to watch where they went on the assembly line.
That is a perfect way to visualize it. So let's look at the RSK cells first, our pristine factory.
Okay.
The flood of heavy glutamine hits the clean conveyor belt. The engine runs forward exactly as it's designed to do in the normal oxidative pathway. The factory burns the fuel, creating this massive spike in energy production.
Right.
But remember, they are also being hit with chemotherapy at the same time. So this hyperactive metabolic state pushes the cell over the edge. It essentially burns itself out. The oxidative stress gets way too high, and the cell undergoes apoptosis.
It dies. Okay, so a clean engine running too fast and burning out under stress makes total sense. Now, explain the broken factory. What happened to the neon green carbon in those KRAS cells?
Well, the KRAS factory has that mutated complex I, right? The forward conveyor belt is physically blocked at step 1, so the glutamine hits a complete dead end.
It can't go forward.
Exactly. But here is the biological marvel of it all. Because there is so much glutamine flooding into the system and it can't go forward, the immense chemical pressure forces the assembly line to run in reverse.
Wait, the engine runs backward. Is that even physically possible in a cell?
Biologically, yes, it is. It's a well-documented survival mechanism called reductive carboxylation. Because the normal oxidative cycle is blocked by those damaged mitochondria, the chemical reactions actually flow in the opposite direction.
That's insane.
And when you run this specific metabolic pathway in reverse, you don't produce normal energy. The mass spectrometer revealed these massive buildups of heavy carbon-13 labeled byproducts.
Byproducts like what?
Specifically, molecules called M+5 citrate and M+3 fumarate.
Okay, let's break those terms down so we don't lose anyone.
Well,
what is an M+5 citrate and why does a tumor even want it?
The M+5 simply means the mass spectrometer detected a citrate molecule containing exactly 5 of those heavy carbon-13 atoms we traced from the glutamine.
Oh, I see. So it proves the citrate was built directly from our flooded fuel.
Bingo. Now, normally, citrate is broken down to make energy. But when the cycle runs in reverse, the cell accumulates it and actually ships it out of the mitochondria.
To do what with it?
To use it as a structural building block. The tumor uses the citrate to synthesize lipids, which are fats and incredibly powerful antioxidants.
Wait, hold on. Let me get this straight. So the blocked assembly line reverses itself. And instead of trying to burn the fuel, it uses the raw glutamine to manufacture antioxidants.
And lipids, yes. You see, chemotherapy drugs often kill cancer cells by causing massive oxidative stress. They literally tear the cell's membranes apart with reactive oxygen species.
Right, they bomb this.
So by running its broken engine in reverse, the KRAS tumor takes that flooded glutamine and builds a massive stockpile of antioxidants to neutralize the chemo's bombs and lipids to fortify its own cell membranes.
Wow.
It is literally taking the raw materials and manufacturing a biological shield. That is why it becomes invincible to the drugs.
Let's pause and really synthesize this, because I feel like this is the crux of the entire paper. Let me know if I have this right.
Go ahead.
You have LSD1, a genetic switch. You turn it off, which unleashes a massive flood of glutamine fuel into the cell. Now, if the cell has healthy mitochondria, it burns that fuel so aggressively that it basically overheats and the chemotherapy destroys it easily.
Oh, exact.
But if the cell has broken mitochondria, it can't burn the fuel. So the chemical pressure forces the metabolic machinery to literally run in reverse. It takes that glutamine, builds an antioxidant shield and thicker walls, rendering the chemotherapy completely useless.
Yes.
The tumor's metabolic identity dictates its ultimate fate.
You nailed it. And the implications of that mechanism are profound,
Wow.
because it proves that the exact same biological input can lead to radically different outputs based purely on the internal environment of the organelle.
So we've uncovered the blueprint of how they survive.
Yeah.
But I guess the next logical step for any scientist is exploitation. If we know the rules of this factory, can we hack the system?
That's the multi-million-dollar question.
Can we force a tumor to change its metabolic identity so the chemotherapy actually works?
Well, the researchers asked that exact question, and I have to say, their experiments to prove it are just elegant. They started with the RSK cells.
The pristine factories that died easily.
Right, the ones highly sensitive to chemo when LSD1 was silenced. They wanted to see if they could intentionally make them resistant. So to do this, they decided to sabotage the factory.
Okay, but how do you intentionally break a cell's mitochondria in a lab?
They used a chemical called rotenone. Rotenone is a really potent inhibitor of that complex I-turbine we talked about.
So they basically jam the turbine themselves.
They basically poison the healthy mitochondria.
Yeah.
And in a parallel experiment, they took a different route and genetically deleted that BNIP3 protein.
Oh, stripping the factory of its waste management system.
Exactly. They introduced the trash buildup artificially.
That is so clever. So what happened?
The moment they crippled the mitochondria, the RSK cells completely changed their identity. When the researchers knocked out LSD1 and flooded them with glutamine, the cells no longer died from the chemotherapy.
No way.
Yeah, they started running their engines in reverse, building those antioxidant shields, and they became completely resistant. They behaved exactly like the KRAS cells.
That is terrifying, honestly, but also an incredible proof of concept. But what about the other direction? Because that's what we really care about for patients. Could they fix the broken KRAS factories and strip away their armor?
They actually could. They took those chemo-resistant KRAS cells and forced them into a metabolic corner. You see, normally, cancer cells love to consume glucose sugar, basically so they can survive without using their mitochondria much.
Right, sugar is easy energy.
But the researchers removed glucose from their diet entirely and replaced it with a different type of sugar called galactose.
Okay, why galactose? What does that do?
Because a cell cannot process galactose efficiently without utilizing its mitochondrial respiratory chain. It literally forces the cell to rely on oxygen and its mitochondria to survive.
Oh, wow.
And in another test, they introduced a specific mutation called KRAS-J12D, which unexpectedly triggers the cell to clear out its damaged mitochondria on its own.
So they essentially forced the broken factory to clean up the trash on its floor and turn its forward conveyor belts back on.
Exactly. And the results were immediate. The moment the KRAS cells were forced to use a healthy, forward-running respiratory pathway, their chemo resistance vanished completely.
Just gone.
Gone. When LSD1 was knocked down, the glutamine flood ran forward instead of backward. The cells couldn't build their antioxidant shields anymore, and the chemotherapy destroyed them.
They successfully hacked the tumor's metabolic identity.
They did.
The biology here is flawless. But I have to play devil's advocate for a second. We have to bridge the gap between, abstract science and real medicine. Because hacking a cell in a flat plastic petri dish is one thing, but a living, breathing tumor inside a pancreas it's a 3D fortress. Did they test this mechanism in a model that actually resembles clinical reality?
They did, and this is where the research becomes highly translational. They moved from those 2D cell lines to 3D organoids.
Organoids, let's explain what those are.
Sure. An organoid is basically a miniature three-dimensional tumor grown in a lab matrix, derived in this case from mice. It closely mimics the complex architecture, the oxygen gradients, and all the cell-to-cell interactions of a real pancreatic tumor in a body.
Okay, so a much better simulation.
A much better test. They used an RSK-subtype organoid model called KPC1 and a KRAS-subtype model called KPC2.
And they didn't just use genetic knockdowns this time, right? Did they use actual drugs?
They use ORY-1001. Now, this is a clinical-grade pharmacological inhibitor of LSD1. It's a real drug that exists in the medical pipeline right now.
Okay, so real drug, 3D model. They applied ORY-1001 alongside standard chemotherapy to these miniature tumors. Did the factory analogy actually hold up in 3D?
Flawlessly. In the KPC1 organoids, the ones with the healthy mitochondria, the ORY-1001 synergized beautifully with the chemotherapy. The miniature tumors shrunk significantly more than with just the chemo alone.
And the broken ones?
In the KPC2 organoids with the broken mitochondria, the ORY-1001 actively antagonized the chemotherapy.
It made it worse.
Much worse. The tumors actually grew more robustly, completely shielded from the chemo's toxic effects by those antioxidant walls.
It's incredibly rare to see a biological mechanism hold its integrity from the molecular weight of a single carbon atom on a mass spectrometer all the way up through complex 3D tissue models.
It's extremely rare, and I think it speaks to the fundamental truth of the mechanism. Metabolism isn't just a byproduct of cancer, it is the core driver of its survival.
Let's bring all these complex threads together for everyone listening. We started with LSD1, a protein heavily linked to deadly pancreatic cancer. The instinct in medicine is to eliminate it. But this deep dive proves that LSD1 is not a simple on-off switch for a tumor's survival.
Not at all.
It is a metabolic traffic cop. It holds back a flood of glutamine. If you remove the cop, the fuel floods in. And how the tumor reacts to that fuel depends entirely on the health of its mitochondrial factory.
Which fundamentally changes how we must approach precision medicine in the future.
How so?
Well, think about it. If a patient comes into the clinic with an RSK-subtype tumor, we know their mitochondria are functional. For that patient, combining an LSD1 inhibitor like ORY-1001 with chemotherapy could be a devastatingly effective combination.
It would overload their system and kill the cancer.
Right. But if a patient comes in with a KRAS-subtype tumor, which is characterized by mitochondrial dysfunction,
then giving them an LSD1 inhibitor alongside chemo might actively build armor for their cancer. We'd literally be helping the tumor survive.
Exactly. For those patients, this research points to a completely different therapeutic target. Instead of targeting LSD1, we might need to target GLS2 directly.
Oh, the enzyme that processes the glutamine.
Yes. If we can block GLS2, the enzyme that converts glutamine into fuel in the first place, we starve that reverse running assembly line before it even starts. They can't build their antioxidant shields, and the chemo can finally do its job.
Man, it's the ultimate lesson in context, isn't it? The exact same drug can be a cure or a curse, depending entirely on the metabolic engine it interacts with. We really can't just read the DNA blueprint anymore. We have to inspect the factory floor.
We do. And the future of oncology lies in that intersection, mapping not just the genome, but the metabolome, and understanding how they communicate with each other.
Well, I want to leave you, the listener, with a lingering thought to mull over today. We just spent this time watching how forcing a tumor to change its diet, its metabolic flow, determined whether a cutting-edge drug worked perfectly or failed completely. It makes you look back at the whole history of oncology with a completely new perspective. I mean, think about it. How many failed cancer drugs are sitting in pharmaceutical graveyards right now?
So many.
Drugs that were thrown out because they didn't show universal success in broad clinical trials. How many of those abandoned compounds might have actually worked flawlessly if we had just known to administer them to the exact right metabolic subtype?
It's staggering to think about. We may already possess the chemical keys to defeat certain cancers. We just haven't figured out which metabolic locks they actually fit into yet.
A profound thought to end on. Thank you all for joining us for this deep dive. Until next time, keep questioning the surface and always look under the hood.